It's a Saturday morning in the 1930s, right in the heart of downtown Manhattan. But you are not surrounded by towering Art Deco skyscrapers or, the loud mechanical roar of early cars.
Well, it's a very different vibe.
Exactly.
Yeah.
You are standing inside the thick circular stone walls of Castle Clinton in Battery Park, which back then housed the original New York Aquarium.
It was such a fascinating building for an aquarium, too.
Oh, totally. It's this cavernous, dimly lit, echoing space. The air in there is incredibly heavy and damp. It smells strongly of brine and wet concrete and cold iron.
I can almost smell it, that very distinct indoor aquatic smell.
Yeah, you know the one. And the only sounds are the echoing footsteps of visitors on the wet floors and this continuous rhythmic, almost heartbeat-like thumping of the massive water pumps.
Just cycling millions of gallons straight from the harbor.
Exactly. And standing in that dim light, completely alone in a massive crowd, is a 9-year-old girl.
She is just completely still, right? Totally separated from the bustling city outside those thick stone walls.
Yes. Her face is pressed so firmly against the thick, scratched glass of a massive tank that her breath is literally fogging up the surface.
Oh, I love that image.
And most of the other people walking past this specific tank are reacting, well, viscerally. They're pointing, shrinking back, grabbing their kids' hands.
Because inside that tank, swimming in endless, silent, powerful circles, is a shark.
Right. To the adults around her, this animal is an absolute monster, a nightmare pulled straight up from the dark abyss.
But this little girl is leaning closer.
Yeah, she is absolutely mesmerized by the sleek, frictionless power of this animal.
It really is a beautiful, haunting image, and it captures the entire soul of our story today. Because that 9-year-old girl staring through the glass was Eugenie Clark.
The legendary shark lady.
Right, that was the nickname the public eventually gave her.
Yeah.
But her legacy is vastly larger than just, a catchy newspaper headline.
Oh, without a doubt. Today, we are exploring the life of a true scientific pioneer.
She didn't merely observe the ocean. She fundamentally dismantled and rebuilt how humanity views marine predators.
And she shattered those incredibly rigid, suffocating glass ceilings of mid-century science, right?
Absolutely. And she revolutionized the very methods of underwater field exploration along the way.
When I try to picture the origins of great naturalists, my mind usually goes to someone growing up wandering through pristine ancient forests.
Right. Or a kid exploring rugged tide pools on some wild remote coastline.
Exactly. But here's a girl whose entire wilderness was a glass box in the middle of a concrete metropolis.
The contrast is just striking. Her wilderness was completely contained. It was artificially curated for public display, surrounded by millions of tons of concrete.
Yeah.
Yet her imagination was completely uncontained. When she looked through that scratched aquarium glass, she didn't see a cage.
No, she saw the vastness of the entire ocean, and she saw a creature of absolute biological perfection.
I want you to hold on to that image as we take this journey. Imagine what it requires of a person to look at a creature that your entire culture tells you is a mindless, terrifying monster.
And instead, to see a completely misunderstood marvel. To look at a nightmare and see a miracle of evolution.
That unique ability to filter out cultural fear and replace it with curiosity is so deeply rooted in her upbringing.
So true. To understand the armor she wore later in life, we really have to start with her childhood in 1920s and 30s New York.
Right. So Eugenie was born on May 4th, 1922, and her life was marked by a profound loss almost immediately.
Her American father, Charles, passed away before she was even 2 years old.
Leaving her to be raised by her mother, Yumiko, who had emigrated from Japan alongside her grandmother.
And eventually, Yumiko married a Japanese restaurant owner named Masatomo Nobu, bringing a new dynamic to their home.
Right. But in those early years, it was a household defined by a powerful, hardworking single mother.
Yumiko worked grueling hours just to keep a roof over their heads. She managed a newsstand right in downtown Manhattan.
And Saturday was a busy workday for her. She desperately needed a safe, free place for Eugenie to stay.
And that safe place ended up being the old aquarium. The institution essentially functioned as her weekend babysitter.
Which is amazing. But the most crucial element here is the cultural lens Yumiko provided her daughter.
Because mainstream American culture in the early 20th century possessed this deep existential dread of the ocean, right?
Oh, completely. The sea was viewed as a dark, violent abyss. Maritime folklore was dominated by tales of man-eating leviathans.
Right, and sharks were universally considered the ultimate evil of the natural world.
Exactly. But Yumiko gave Eugenie a profoundly different perspective. a traditional Japanese reverence for the sea.
The ocean wasn't a place of horror. It was a place of bounty. It was the source of life.
Yeah, in an island nation like Japan, the ocean dictates survival. It shapes the diet, the mythology, the art, and the spirituality.
So Yumiko taught Eugenie to look at marine life with profound respect.
Right. When Eugenie looked at that shark, she wasn't looking through the lens of Western fear. She was looking through the lens of her mother's cultural heritage.
Which is such a beautiful foundation. But man, the reality of her daily life outside that apartment was incredibly harsh.
The isolation she faced was severe. She was the only student of Japanese descent in her entire school in Queens.
Just picture that cafeteria dynamic. She opens a lunchbox and instead of, I don't know, a standard ham sandwich, she brings out meals packed with seaweed and fish cakes.
And her classmates, growing up in a very insulated era, were completely bewildered.
And children can be incredibly cruel when faced with something unfamiliar.
Oh, absolutely. But the cruelty went far beyond simple playground teasing.
Yeah.
There was active, targeted prejudice.
Yeah, there's that deeply telling story from her elementary school years that illustrates this perfectly, right?
Yes. So Eugenie was a highly talented artist, and she poured her love of the ocean into this beautiful, detailed drawing of marine life.
And the school recognized its quality. They awarded her a prize, proudly hanging the artwork on the wall for everyone to see.
Until someone came along and maliciously wrote a vicious racial slur right across the center of her drawing.
It's just awful. They defaced the thing she was most proud of, attacking her identity in the most public way possible.
It is devastating to imagine a child enduring that kind of targeted hatred.
Most kids would just retreat, maybe try to assimilate or hide their passions.
But she hardened her resolve. If the outside world was going to be hostile, she decided to construct her own world.
Right. She literally transformed her tiny New York bedroom into a functioning biological laboratory.
She acquired dozens of glass tanks, stacking them on every available surface, filling them with guppies, swordtails, and whatever small aquatic life she could find.
She was instinctively developing the skills of a behavioral ecologist before she even knew the term existed.
Exactly. She spent hours quietly observing how they moved, how they established social hierarchies, and how they reproduced.
And while her peers at school alienated her, she found intellectual community in the most unexpected place imaginable.
The men hanging around Battery Park.
Yeah. She affectionately called them the Bowery Bums.
On bitterly cold or rainy days, these homeless men would seek shelter inside the warm, cavernous halls of the aquarium.
And Eugenie, this tiny, endlessly curious girl, just struck up conversations with them.
And as it turned out, some of these men were highly educated. They were individuals who had simply been crushed by the economic devastation of the Great Depression.
So they essentially became her first scientific colleagues.
They did. They would walk the perimeter of the tanks with her, pointing out anatomical details, sharing complex biological facts, and encouraging her curiosity.
They provided the validation that her classmates denied her.
Right. And alongside those men, her imagination was being stoked by the legendary explorer William Beebe.
Oh, Beebe. At that time, he was making these unprecedented vertical plunges into the pitch-black depths of the ocean inside a device called a bathysphere.
Which is an incredible piece of early tech.
If you can call it that. It's essentially just a hollow, heavy iron cannonball with a tiny quartz window dangled thousands of feet into the abyss on a single metal cable. It sounds absolutely terrifying.
It was incredibly dangerous. But Eugenie devoured every written count of his descents. She made a silent promise to herself that she would follow him into the deep.
Looking at those formative years, the racist attacks at school, the loss of her father, the poverty, the solace found with homeless men in a damp fort, you really see the forging of an incredibly tough human being.
Oh, she openly acknowledged the impact of those years. She credited those painful experiences with shaping her adult personality.
She said they made her aggressive, spunky, and fiercely defensive, right?
She forged a suit of armor, and it was an armor she would absolutely need.
Because transitioning from a bright child with a bedroom full of guppies to a recognized professional in the mid-20th century scientific establishment meant running headfirst into a towering institutional brick wall.
She certainly had the academic credentials. She earned her Bachelor of Arts in Zoology from Hunter College in 1942.
And then pushed forward to earn a master's degree in 1946 and eventually a PhD in 1950 from New York University.
And her PhD work was this brilliant full-circle moment. She studied the live-bearing reproduction mechanisms of the exact same species of small fish she had kept in her childhood bedroom.
I love that so much. But securing those degrees was like climbing a mountain during an avalanche.
The landscape of academia in the 1940s was profoundly homogeneous. It was overwhelmingly white and overwhelmingly male.
Women weren't just passively overlooked in the hard sciences, they were actively, aggressively discouraged from participating.
There was one specific roadblock she faced that just makes my blood boil.
Oh, the Columbia incident.
Yeah. After finishing her undergraduate degree, she applied to Columbia University for a master's program. She secures an interview with a highly prominent, influential professor.
And she sits down, a brilliant, proven, eager young zoologist, and this man looks her dead in the eye and tells her he will absolutely not support her application.
His reasoning was breathtakingly condescending. He stated that even if she successfully completed the rigorous program, she would inevitably just get married, have a house full of children, and completely waste the university's time, resources, and funding.
He reduced her massive intellect and her lifelong passion to a flat domestic stereotype.
It's infuriating.
Imagine training your entire life for a sport, proving you're the most capable athlete on the field, and when you finally arrive at the championship game, the officials ban you from playing. Not because you lack the skill, not because you don't know the strategy, but simply because they do not like the uniform you were born in.
It was a brutal rejection. And unfortunately, her gender was only one of the weapons used against her. Her Japanese heritage was another.
Right, because in the years immediately following World War II, the United States was steeped in a toxic, lingering anti-Japanese paranoia.
Even though she was born in New York, raising an American flag, eating hot dogs.
Yeah, her citizenship did not shield her from suspicion at all.
In 1947, the U.S. Fish and Wildlife Service, recognizing her exceptional talent, invited her to conduct biological field work in the Philippines.
Which is a monumental opportunity for a young scientist to establish her career on an international stage.
Exactly. So she packs her bags and travels as far as Hawaii, which was the halfway point of her journey.
And then the federal government steps in.
She was detained by the FBI solely because of her Japanese last name and her ancestry. Wartime prejudices caused the authorities to revoke her clearance.
So she was blocked from traveling to the Philippines and forced to return home, her massive career opportunity completely crushed.
The door wasn't just closed, it was slammed in her face and deadbolted.
But this is exactly where that spunky and defensive armor kicks in. She completely refuses to quit.
She eventually finds a lifelong mentor back at NYU, Charles M. Breder Jr., a man who actually evaluates her based on her intellect.
And with his backing, she secures a position at the Scripps Institution of Oceanography in California.
And this is where the story takes a thrilling turn, because she finally gets to submerge herself in the open ocean.
But we have to remember, this was long before the invention of the sleek, lightweight scuba gear we see today.
Oh, underwater exploration in the late 1940s was archaic and physically punishing. She had to learn using the traditional heavy copper helmet gear.
Paint a picture of what that actually involves physically, because it sounds like a medieval torture device.
It essentially is. You are wearing a massive, rigid canvas suit. A solid copper helmet weighing upwards of 60 pounds is bolted to a heavy metal breastplate resting on your collarbones.
60 pounds just resting on your chest.
Yeah. And you are completely dependent on a rubber hose connecting the top of your helmet to a manual air pump on the surface. Someone on a boat has to constantly manually crank that pump to send you air.
So you have zero peripheral vision, you are carrying half your body weight in metal, and your life is entirely in the hands of the guy turning a wheel on a boat.
And the margin for error is absolutely zero. During one of her early descents with this gear, a malfunction occurred, and water began rapidly flooding into her helmet.
Oh my gosh.
She nearly drowned in the dark, trapped inside that heavy copper bell.
Yet the sheer physical terror of near death didn't deter her any more than the academic gatekeepers did.
She just shakes it off. And in 1947, she finally makes it to the Pacific, traveling to the islands of Palau in Micronesia.
And this location is a huge turning point for her methodology, right?
Completely. Most Western scientists of that era would fly into a remote location, set up their sterile equipment, and completely ignore the people who actually live there.
But Eugenie takes the opposite approach. She pairs up with a local Palauan fisherman named Siakam.
This decision demonstrates a profound scientific humility. The Western academic establishment considered indigenous knowledge to be, anecdotal or unscientific.
But Clark immediately recognized that Siakam possessed a generational understanding of the reefs.
He knew the precise behavioral patterns of the fish, the shifting of the seasonal tides, and the hidden locations of the most elusive species.
She treated him not as an assistant, but as a senior colleague, and her scientific output was exponentially better because of it.
And it is in the crystal clear waters of Palau that she experiences the moment she had been dreaming of since she was 9 years old.
She has her very first encounter with a wild shark.
The scratchy aquarium glass is finally gone. She is suspended in the open water, completely exposed, and a shark materializes from the blue distance and approaches her.
The human nervous system is practically hardwired to panic in that scenario. The sheer size and predatory design of the animal naturally triggers a primal fight or flight response.
But she felt no fear at all. She later described the moment as one of pure, unadulterated awe.
She was finally sharing the water with the perfect creature she had admired her entire life.
That singular moment in Palau really solidified her destiny. The traditional male-dominated institutions of the American East Coast were constantly trying to barricade her path, but the ocean itself had no gates.
She realized that to succeed, she needed to carve her own route into an extreme, remote environment, a place where very few Western scientists, let alone a young woman of color, had ever dared to venture.
She sets her sights on the Red Sea. Supported by a prestigious Fulbright scholarship in 1950, she travels to the Al-Gardaqa Marine Biological Station in Egypt.
The Red Sea is not just another body of water. It's one of the most geologically and biologically extreme environments on the planet.
I'm glad you brought that up. Explain why it is so extreme, because understanding the environment is key to understanding your discoveries there.
Okay, so it begins with tectonic violence. Millions of years ago, deep mantle convection caused the African and Arabian tectonic plates to begin ripping apart.
Right, creating a massive, deep rift valley in the Earth's crust.
Eventually, the Indian Ocean breached the natural barriers and flooded the searing desert canyon, creating the Red Sea.
So it is essentially a flooded gorge surrounded on all sides by hyper-arid, baking desert.
Exactly. And the geography creates intense abiotic stress. Because it's surrounded by desert, there are virtually no major rivers flowing into it to supply fresh water.
Meanwhile, the blistering sun beats down, causing an astronomical rate of evaporation.
And when ocean water evaporates, the H2O molecules rise into the atmosphere, but the salt is left behind.
Making the remaining water incredibly dense and highly saline. It sounds completely hostile to life.
It forces life to adapt in extraordinary ways. Due to this extreme heat, the high salinity, and the sea's relative isolation from the broader Indian Ocean, the Red Sea became a massive engine for endemism.
Endemism meaning species that evolve in one highly specific location and exist literally nowhere else in the universe.
Precisely. It is an isolated evolutionary laboratory. And Eugenie arrives in Egypt right as the entire global discipline of marine biology is undergoing a massive, painful paradigm shift.
For decades prior, if a zoologist wanted to study a fish, they usually went down into the dusty basement of a natural history museum.
Right. They pulled a dead, gray, chemically preserved specimen out of a glass jar of formaldehyde.
They counted the scales along its lateral line, measured the length of its pectoral fins, assigned it a Latin name, and considered the job done.
It was essentially cataloging biological stamps. But Eugenie wanted to see the living machine in motion.
She wanted to observe how these animals interacted within their complex ecosystems, how they hunted, how they evaded capture, how they communicated.
And her observational skills lead her to one of the most astounding discoveries of her career. She finds a little flatfish called the Moses sole.
Known scientifically as Pardachiris marmoratus.
On the surface, this fish sounds like a complete myth.
It looks completely unassuming, like a small, mottled brown flounder just blending into the sand.
But Clark noticed something extraordinary about its interactions with predators. She discovered that this flatfish secretes a milky, naturally occurring chemical substance from a series of specialized pores running along the base of its fins.
And this milky substance is a highly potent, targeted shark repellent.
It is a chemical weapon deployed against the apex predator of the reef. Clark observed and documented the mechanism.
If a shark attempts to bite the Moses sole, the chemical immediately interacts with the shark's nervous system and musculature.
It completely disrupts the motor neurons, causing the shark's massive jaws to physically freeze open in a state of tetany.
The shark is literally paralyzed mid-bite.
Exactly. It completely disables the predatory response, allowing this tiny, slow-moving flatfish to casually swim away unharmed while the shark struggles to close its mouth.
The biological ingenuity is just staggering.
Now, I know later military and medical testing revealed that this specific chemical couldn't be easily synthesized or stabilized into a practical, sprayable shark repellent for human use.
Right, but the mere discovery of the biological mechanism proving that a tiny fish had evolved a chemical defense to stop a great predator in its tracks made international headlines.
Her success came from her willingness to look where others didn't. Most underwater explorers in the Red Sea were entirely captivated by the massive, colorful, vibrant coral reefs.
And the reefs are loud, busy, and visually overwhelming.
But Clark often turned her back on the coral. She would spend hours hovering silently over the seemingly barren, empty stretches of sandy seafloor.
Which most people just view as the underwater desert you have to cross to get to the next pretty reef.
But she knew the sand held its own secrets. Her patience paid off when she spotted a tiny, needle-like fish that had evolved a fascinating evasion tactic.
It would hover above the bottom, and at the first sign of danger, it would rapidly burrow backward, tail first, deep into the sand until it completely vanished.
It was a completely unrecorded species. Because she discovered it, she earned the right to name it, calling it Trichinotis nicky, the Red Sea sand burrower.
She named it after her youngest son, Nicky.
She eventually had 4 children, Hera, Aya, Tok, and Nikki, and she routinely brought them along on her international expeditions later in life.
She spectacularly proved that condescending Columbia professor wrong. She didn't have to choose between a family and a groundbreaking scientific career. She seamlessly integrated them.
Her work in Egypt cemented her entire biological philosophy. She didn't view the ocean as a collection of isolated specimens. She treated the sea as a single, massive, breathing organism.
She respected the apex sharks, the tiny sand-burrowing fish, and the complex chemical warfare holding the balance of power together.
To share this philosophy, she wrote a detailed account of her experiences in Egypt.
Published in 1953, her memoir, Lady with a Spear, became a massive, unexpected global bestseller.
It translated the dry, academic world of marine biology into a thrilling, accessible, romantic adventure for the general public.
And crucially, the success of this publication caught the attention of some incredibly wealthy, highly influential people back in the United States.
This success pulls her from the ancient hypersaline waters of the Red Sea and drops her directly onto the humid, swampy, mosquito-filled coast of 1950s Florida.
2 wealthy philanthropists, Anne and William H. Vanderbilt, read her account and were entirely captivated by her vision and her unyielding grit.
They reached out to her with an unprecedented offer. They wanted to financially back the creation of a brand new marine laboratory, with her as the founding director.
In January 1955, Eugenie Clark officially opened the Cape Hayes Marine Laboratory in Placida, Florida.
When someone mentions a scientific laboratory funded by the immense wealth of the Vanderbilt family, a very specific image comes to mind.
Oh yeah, you picture a sprawling multi-acre campus, massive glass atriums, sterile white rooms, and hundreds of technicians in pressed lab coats.
The reality was slightly less glamorous. The Cape Hayes Marine Laboratory was, in fact, a single 12 by 20-foot wooden shed.
A wooden box on the edge of a swamp.
Literally. A simple wooden shack equipped with a basic plumbing sink, some rough wooden shelves for holding specimen jars, and a single 21-foot wooden boat named Dancer tied up to a small, creaky dock.
That was the entirety of the facility.
It's incredibly humbling to realize that one of the great scientific revolutions of the 20th century didn't begin in a billion-dollar facility, but in a wooden shack smelling of formaldehyde and dead fish.
And her initial staff consisted of exactly one person. She hired a seasoned, local Florida commercial fisherman named Beryl Chadwick.
Once again, she demonstrates her absolute respect for practical, lived experience.
Right. She didn't import a team of Ivy League graduate students who only knew oceanography from a chalkboard. She hired the man who actually knew the waters of the Gulf Coast.
Chadwick knew the shifting sandbars, he knew how the tidal currents operated, and most importantly, he knew how to safely track and capture large sharks.
They worked side by side in the searing Florida heat. To collect the smaller specimens needed for the lab's various studies, they relied on a highly technical, complex method known as the glass jar technique.
Which, knowing her practical nature, I assume literally means swimming around and scooping up fish with an empty mayonnaise jar.
That is exactly what it means. It was grassroots, bootstrap science in its purest, most exhausting form.
But despite the incredibly humble facilities, the quality of the scientific output was undeniable. Her reputation grew astronomically.
Biologists and medical investigators from all over the world began traveling to this tiny, sweltering Florida town just for the chance to collaborate with her.
The operation expanded so rapidly that by 1960, they simply outgrew the shed and had to move to a much larger, more robust facility on Siesta Key.
And then, in 1967, the institution underwent another major transformation. It was officially renamed Mote Marine Laboratory.
The renaming was a gesture of immense gratitude toward William R. Mote, a highly successful transportation executive who had dedicated his personal fortune, his land, and countless hours of his own time to supporting the sea and expanding the laboratory's operational capabilities.
I want to linger on that name change for a moment because it speaks volumes about Eugenie Clark's character.
She founded this place. She built its global reputation from absolute scratch in a tiny wooden shed.
But the massive world-renowned laboratory doesn't bear her name.
It reveals a profound lack of ego. She was never driven by the desire for personal glory or fame.
She recognized a pragmatic truth. The immense financial and logistical support William Mote provided would ensure the laboratory's survival, growth, and independence for generations to come.
She prioritized the continuity of the scientific work and the protection of the ocean far above having her own name carved into the building.
And today, Mote Marine Laboratory stands as one of the premier independent marine research institutions on the planet.
And it was at this very lab, starting way back in the humble Cape Hayes shed days, that the most famous, world-altering experiments of her entire career took place.
The experiments that would permanently dismantle the myth of the mindless eating machine.
The catalyst for this monumental shift in animal behavioral science was surprisingly mundane. A medical investigator studying cancer reached out to Clark with a practical request.
He asked if she could capture some wild sharks and send him their livers for his cellular studies.
To keep the sharks alive before sending the organs to the medical investigator, Clark and Beryl Chadwick had to construct a live holding pen out in the bay.
They drove heavy wooden pilings deep into the muddy bottom and securely fenced off a large section of the tidal water.
And once she had these live, powerful sharks swimming around in a contained, semi-natural environment right off her dock, her mind began to race with possibilities.
Because in the late 1950s, the established scientific dogma regarding the intelligence of sharks was incredibly dismissive, almost insulting.
The prevailing consensus among the world's leading marine biologists was that sharks were evolutionary dead ends when it came to cognition.
Right, because sharks are cartilaginous fish, meaning their entire skeletal structure is made of flexible cartilage rather than calcified bone.
Yeah.
They were viewed as ancient, primitive leftovers from a bygone era.
The academic world widely believed their brains were incapable of anything beyond base instincts. Smell blood in the water, swim forward, bite down.
They were viewed as the ultimate biological automatons, mindless, instinct-driven robots.
But Eugenie watched them navigating her wooden pen, observing their subtle behaviors, and she suspected the entire global establishment was fundamentally wrong.
She decided to give these so-called mindless machines an intelligence test.
This decision led to her landmark 1959 publication in the journal Science titled Instrumental Conditioning in Lemon Sharks.
Let's break down the mechanics of this experimental design, because figuring out how to administer an IQ test to a massive aquatic predator is a puzzle in itself.
The foundation of her approach relied on operant conditioning, sometimes referred to as instrumental conditioning.
To clarify, this is a step beyond simple Pavlovian conditioning, where an animal just associates a random sound with food.
Operant conditioning requires the animal to understand agency. They must learn that their specific, deliberate physical action directly results in a specific consequence or reward.
So she turns this fenced-in bay pen into a giant underwater Skinner box.
She constructs this clever mechanical contraption featuring a bright white wooden square target that is lowered beneath the surface. Rigged just behind this target is an underwater bell.
She initiates the trials using lemon sharks, known scientifically as Megaprion brevirostris, and nurse sharks, Genglima seratum.
In the earliest stages, she and her team simply dropped food into the water directly next to the white target to draw their attention.
But slowly, methodically, they began to shape the required behavior.
The shark eventually figures out that to get the food, it can't just passively wait. It has to actively swim up to the contraption and physically press its snout against the white wooden square.
The pressure against the target triggers the bell to ring, and the sound of the bell signals the team on the dock to drop a piece of fish into the water as a reward.
The mere fact that the sharks successfully learned this basic mechanical association press target, hear bell, get food, was enough to shock the established scientific community.
But Clark knew they were capable of more, so she drastically increased the cognitive difficulty of the task.
She wanted to test the limits of their flexible thinking. So she started moving the food reward location further and further away from the wooden target.
Eventually, the shark would have to press the target, hear the ringing bell, and then immediately turn and navigate to a completely different, distant area of the pen to retrieve the food.
And to make it even harder, she imposed a strict time limit. They only had 10 seconds to make the correct turn, navigate the space, and reach the drop zone before the food was withheld.
This requires a highly sophisticated suite of cognitive abilities.
It demands spatial awareness to map the layout of the pen, working memory to hold the goal in mind, and the executive function to link a chain of distinct events into a cohesive, successful strategy.
And the sharks mastered it flawlessly.
But working memory is one thing. What about long-term retention? We constantly hear that ridiculous myth that goldfish have a 3-second memory.
Right. What is the long-term memory capacity of a cartilaginous fish?
Clark designed a brilliant, naturally occurring memory test to answer that exact question.
When winter arrived on the Florida coast, the ambient water temperatures in the shallow bay dropped significantly.
As cold-blooded animals, the sharks' internal metabolisms naturally slowed down in response to the chill, and they entered a period of dormancy where they completely stopped eating.
For 10 solid weeks, the team completely halted the conditioning experiments. The white targets were pulled from the water. The bells were silenced.
10 entire weeks. 2.5 months of absolute silence and no reinforcement.
When the spring sun finally warmed the waters of the bay, the sharks' metabolisms ramped back up. Clark took the white wooden target and lowered it back into the pen.
The sharks hadn't seen, touched, or heard the mechanism in over 2 months.
What was their immediate reaction?
The very moment the white square breached the surface of the water, the sharks swam directly toward it, pressed their snouts firmly against the wood, rang the bell, and immediately banked to swim toward the distant reward zone.
They remembered the exact mechanical sequence and spatial map of the system.
The long-term retention is incredible, but my absolute favorite detail from this entire era of her work involves the female sharks.
Oh, yes. The cheating females perfectly illustrate just how adaptable their intelligence really is.
It is a phenomenal example of high-level behavioral adaptation and social observation. In the holding pen, Clark had a mixed population of male and female sharks.
The males were highly active participants, eagerly swimming up, aggressively hitting the target, and then rushing across the pen to consume the food.
But the team observing from the dock noticed that some of the female sharks realized they didn't actually need to expend the energy required to hit the target.
They observed the mechanics and completely hacked the system.
They deduced that the sound of the bell was the true indicator that food was about to drop in a very specific location.
So instead of swimming all the way over to the target, the females would lazily, quietly circle the exact spot where the food was going to fall.
They would simply wait for the males to do the hard work of swimming over, hitting the target, and ringing the bell.
And the split second the food hit the water, the waiting females would snatch it up before the males could even turn around to claim their prize.
They manipulated the males into doing the labor. It is identical to that moment when you realize your dog hasn't just learned to sit to get a treat, but has actually figured out how to manipulate your routine to force you to open the treat jar.
It demonstrates an incredibly high level of environmental awareness and social opportunism.
While it is deeply amusing, it's important to clearly define what we mean by intelligence in this biological context.
When we discuss shark cognition, we're not suggesting they possess human-like reasoning or that they're doing complex algebra in their heads.
They aren't pondering the philosophical meaning of the bell.
Exactly. Cognitive flexibility in an apex predator means the animal possesses the dense neurological machinery required to process complex, shifting sensory data from its environment.
To form lasting associative memories, and to alter its future behavior on the fly to maximize its chances of survival and minimize energy expenditure.
Clark proved that sharks possess highly evolved, specialized intelligence perfectly calibrated to dominate their ecological niche.
They aren't mindless, primitive machines. They are magnificent, highly adaptable learners.
This realization triggers a massive domino effect. If the entire global scientific establishment was dead wrong about the brain capacity of sharks, what other fundamental facts were they completely wrong about?
That exact line of questioning brings us to another massive history-altering paradigm shift in the 1970s. The mystery of shark respiration and the discovery of the sleeping sharks.
Set the stage for this mystery, because it challenges everything we thought we knew about how sharks survive.
For decades, there was an ironclad, unquestionable biological rule regarding streamlined, pelagic sharks, the torpedo-shaped species that constantly patrol the open ocean.
The rule was simple and absolute. If they stop swimming forward, they will immediately suffocate and die.
What was the anatomical justification for that belief? Why would a fish have to constantly move just to breathe?
To understand the error, we have to compare the anatomical mechanics of a standard bony fish, like a large grouper or a snapper, to the anatomy of a shark.
If you examine the side of a bony fish's head, you will see that their delicate gills are protected by a hard, articulated bony plate called an operculum.
Furthermore, the skull of a bony fish is an incredibly complex, highly jointed mechanical structure packed with powerful throat and jaw muscles.
They have the physical hardware to actively pump water, like a biological bellows.
Precisely. A bony fish can rest completely motionless on the ocean floor, open and close its mouth, and use those powerful muscles to actively draw oxygen-rich water in, force it over the gills to extract the oxygen, and push the deoxygenated water out through the operculum.
Now contrast that with the head of a shark.
Sharks just have those 5 to 7 iconic slits sliced into the side of their neck.
5 to 7 independent exposed gill slits, completely lacking a rigid bony cover.
And a shark's skull is relatively simple, a fused box of flexible cartilage that lacks the complex mechanical hinges and the powerful, specialized pumping muscles found in bony fish.
Marine biologists of the era often joked that despite their fearsome reputation and massive bite force, sharks suffered from wimpy gills.
Wimpy gills.
Because the musculature surrounding those exposed gill slits is so relatively weak, the scientific consensus held that streamlined sharks were physically incapable of pumping a sufficient volume of water to survive while remaining stationary.
Biologists believe these predators relied entirely on a process called ramjet ventilation.
Meaning they have to constantly swim forward with their mouths slightly open, literally ramming the ocean water over their gills like a jet engine taking in air at high speed.
Ceaseless, lifelong forward motion from birth until death. That was accepted as absolute fact.
Until 1969, when an intrepid underwater explorer named Ramon Bravo discovered something completely impossible inside a series of submerged marine caves off the coast of Isla Mujeres, Mexico.
He swam into these caves and found streamlined sharks lying completely motionless on the rocky floor, just resting there like parked cars.
It defied every established law of marine biology. When Eugenie Clark heard about this impossible phenomenon, she knew she had to witness it firsthand.
Backed by funding from the National Geographic Society, she organized expeditions to Isla Mujeres in 1972 and 1973 to investigate the anomaly.
Imagine the tension of swimming into a dim, enclosed underwater cave, your flashlight cutting through the gloom and seeing the floor completely carpeted with deadly apex predators, all lying together in perfect silence.
It was an eerie, breathtaking, almost supernatural scene. Her close field observations identify the animals as Caribbean reef sharks, Carchahinus perezi.
And as she cautiously approached them, she realized they were neither dead nor suffocating.
Even though their bodies were perfectly still, they were actively, rhythmically pumping water over their gills at a rate of 20 to 28 times a minute.
They were doing exactly what generations of textbooks said their anatomy could not do.
But the bigger question became, why were they doing it in these specific caves? They could theoretically rest anywhere, so what made these caves special?
That became the new driving mystery. Clark brought in specialized instruments to test the abiotic factors of the water inside the cave system, mapping the chemistry of the environment.
She discovered 2 major anomalies. First, the water trapped inside the caves had unusually high levels of dissolved oxygen.
Second, the water had a significantly reduced salinity level compared to the open ocean just a few yards outside the cave entrance.
How does an underwater cave in the middle of the ocean become less salty?
The Yucatan Peninsula is made of highly porous limestone. The reduced salinity was caused by massive freshwater upwellings, underground rivers and springs flowing from the Mexican mainland, seeping up through the porous floor of the cave, and mixing with the heavier salt water.
So you have high oxygen and low salt. How does that explain a floor full of resting sharks?
Clark formulated a series of brilliant ecological hypotheses. The high oxygen content might have provided a chemical assist, making it significantly easier for their wimpy gills to extract the necessary oxygen without the aid of forward motion.
She even speculated that the hyperoxygenated water might give the sharks a sort of mild, narcotic, relaxing physiological high.
An oxygen high for a Caribbean reef shark.
But the reduced salinity offered a much more tangible, practical biological benefit.
Wild sharks are constantly plagued by marine parasites, tiny coat pods, and parasitic leeches that embed themselves deep into the shark's tough skin.
These marine parasites have evolved to survive in high saline environments. They absolutely hate fresh water.
The lower salinity in the caves causes the cells of these parasites to undergo osmotic stress, forcing them to loosen their agonizing grip on the shark's flesh.
The cave is essentially a giant natural deworming station.
Exactly. Clark proposed that these caves functioned as highly specialized cleaning stations.
While the massive sharks lay completely docile and relaxed in the brackish water, smaller, specialized fish called remoras, often known as shark suckers, could safely flutter over the predators' bodies, meticulously picking off and eating all the loosened parasites.
It is a perfect mutualistic relationship. The remoras get a free, concentrated buffet, and the sharks get a deep, therapeutic clean without being bitten.
But it leads to a fascinating behavioral question. Were the sharks actually asleep during this process?
That remains the million-dollar biological question. Sleeping sharks makes for an incredible magazine headline, but biologically speaking, true sleep is defined by profound, measurable shifts in brainwave activity.
To date, no one has successfully managed to attach an EEG machine to the brain of a wild Caribbean reef shark while it rests in a Mexican cave.
However, all the observational data gathered by Clark and others heavily suggest they are not truly asleep.
The underwater explorers noted that as they moved cautiously around the perimeter of the cave, the eyes of the motionless sharks would track them intently, following their every move.
So they're fully conscious, totally aware of their surroundings, and ready to react, but choosing to remain deeply relaxed.
And this incredible discovery sparked a tidal wave of new physiological investigations.
Years later, a prominent biologist named Samuel Gruber conducted rigorous manometry experiments on lemon sharks in a controlled laboratory setting in Bimini.
He utilized a specialized sealed chamber to measure the exact rate of oxygen consumption in the water while the shark was forced to swim against the current versus when it was allowed to rest stationary on the bottom.
Measuring the exact metabolic cost of resting versus swimming.
And he proved something utterly astonishing. Resting motionless on the bottom and actively forcing water over their gills actually requires 9% more oxygen consumption than simply swimming forward and utilizing passive ramjet ventilation.
Wait, so lying completely still is actually harder physiological work for them than swimming?
It's a net energy loss. This revelation shattered everything. It meant they aren't resting in these caves to conserve energy.
Whatever they are doing down there, whether it is getting cleaned by remoras, socializing, seeking a safe refuge from larger predators, or simply enjoying the tactile sensation of the sandy floor, they are making a calculated behavioral choice to expend extra energy to achieve it.
It reveals a level of social and behavioral complexity that the scientific world had never imagined possible for a cartilaginous fish.
Every single time she looked closely, she proved that these animals were exponentially more complicated, adaptable, and fascinating than the dusty textbooks claimed.
And as she grew older, her insatiable curiosity only pushed her to explore deeper, literally into the dark.
In her later years, a time when many decorated scientists might choose to retire to a comfortable desk or a tenured advisory role, Clark fully embraced the final, most punishing frontier of marine exploration: the deep ocean abyss.
She transitions from staring through the thick, scratched glass of the New York Aquarium as a child to staring through the thick acrylic spheres of deep-sea submersibles as an elder stateswoman of science.
Over the course of her lifetime, she conducted more than 200 major field expeditions, and she made an astounding 72 submersible descents.
She plunged into the lightless, freezing depths, sometimes descending as far as 12,000 feet below the surface of the ocean.
The pressure at that depth is incomprehensible, but she found immense joy in the absolute darkness.
Alongside her exploration, she spent decades teaching zoology at the University of Maryland, inspiring entirely new generations of students to look past their fears.
And she became a fierce vocal advocate for marine conservation.
She fought tirelessly to protect the fragile ecosystems she had spent her life exploring, most notably leading the charge to protect the magnificent coral reefs of Ras Mohammed in Egypt, which were under severe threat from commercial destruction.
Her impact on the field was so profound and lasting that the scientific community repeatedly chose to honor her by weaving her name into the very taxonomy of the oceans she loved.
My absolute favorite example of this taxonomic honor occurred recently, in 2018. Biologists were studying deep-sea dogfish sharks in the Gulf of Mexico.
Deep-sea sharks are notoriously difficult to classify visually.
The evolutionary pressures of surviving in the abyss, the total lack of sunlight, the near-freezing temperatures, the crushing hydrostatic pressure are so intense that they force many completely different species to evolve very similar, highly optimized body shapes.
This biological phenomenon is called convergent evolution.
Think of it like trying to build a vehicle to survive a freezing underwater obstacle course. It doesn't matter if Ford, Chevy, or Toyota tries to build it. Eventually, the physics of the obstacle course will force all of them to build something that looks exactly like a submarine.
The environment dictates the shape. Because of this, for a long time, scientists assumed this particular shark in the Gulf was just an isolated population of Squalus mitsukuri, a globally recognized species of dogfish.
But when biologists finally extracted tissue samples and ran modern genetic DNA testing, the sequence revealed something startling.
It was a completely distinct, previously undocumented species. It had been hiding in plain sight in the deep water.
Armed with the genetic proof, they looked much closer at the shark's morphology, its physical structure, and realized it did have subtle but distinct differences. A slightly longer body, a tighter gap between its eyes, and a shorter caudal fin, which is the tail fin.
And having identified this brand new species of deep-sea predator, what did the scientific community choose to name it?
Squalus clarkae, Genie's dogfish.
Having a species named in your honor is the ultimate sign of respect for a taxonomist.
And the honors extended far beyond the insular halls of academia. In 2022, on what would have been the 100th anniversary of her birth, the United States Postal Service issued a forever stamp to commemorate her life's work.
The design of the stamp is visually striking. Created by artist Amanda Fingbodapekia, it features a beautifully rendered collage of photos of Eugenie's face looking out, set right alongside a magnificent image of a lemon shark gliding through the water, taken by Reinhard Derschel.
It is incredibly fitting that her face, alongside the very animal she dedicated her life to understanding and defending, will travel on letters and packages to every corner of the country.
It serves as a miniature monument, ensuring that her legacy and her core message of prioritizing curiosity over fear continues to circulate in the public consciousness.
Eugenie Clark passed away in 2015 at the age of 92. When you step back and view the entire timeline of her life, the scale of her impact is staggering.
She took an animal that humanity had universally agreed was a mindless, evil, instinct-driven eating machine, and through sheer patience and brilliant observation, she proved it was a magnificent, adaptable, highly intelligent creature.
She built a massive, globally respected scientific institution starting from a 12 by 20 wooden shed in the swamp.
And beyond the biological discoveries, she fundamentally changed the face of who was allowed to participate in science.
She navigated a brutal gauntlet of systemic sexism and post-war racism with grace, sharp humor, and an unstoppable, spunky determination that refused to be denied.
As we bring this incredible journey to a close, I want to leave you with one final thought to mull over.
Humanity spent centuries calling sharks mindless monsters simply because we lack the imagination to figure out how to test their intelligence.
Or worse, because we were too terrified to even bother looking closely. We allowed our cultural fear to dictate the boundaries of our understanding.
It raises a profound question about our relationship with the unknown elements of our world.
What other monsters are out there right now, whether they are misunderstood creatures deep in the natural world or marginalized communities within our own society, that are simply waiting for someone with enough patience, enough respect, and enough relentless curiosity to finally understand them? Thank you so much for joining us on Saltwater Stories today. We hope you leave feeling a little more curious and a little less afraid of the wild world around you. Until next time, keep looking closer at the mysteries just beneath the surface.